So thanks a lot, Alex, for the kind introduction, and many thanks for the invitation, because actually this is an amazing conference. I really enjoy being there. So I will try to share with you a few things we are currently doing in Switzerland. First of all, I have no conflict of interest. So we've heard a lot about NGS today, yesterday also actually. And so you all know finding the right variant looks like this in many cases. I would say it's pretty similar to trying to find Switzerland on the map of the world. So this is a paper from 2020, where you see it's colored by the number of, total number of bioinformatics publication. So Switzerland is somewhere in the middle of Europe. But this is because we don't look at it from the right angle. If you look at the very same data, but in this case, you look at the number of bioinformatics publications per capita, so per inhabitants, then Switzerland is the small black or dark red dot in the middle of Europe. So now you all know where Switzerland is standing, basically. Just let me give you a very, very brief introduction about what SIB is. SIB is a special entity, actually. We are a non-for-profit organization, but it's pretty special because we have 800 people, but out of these 800, we have 200 employees, more or less. And the other 600 people, they are not directly employed by SIB, but they are affiliated with SIB, meaning they are spread all over the country in 23 other institutions. So it's quite special, but it's really great, actually, to be part of this, because we cover a lot of activities. Obviously, with 85 groups, you have activities for everyone. And what is really nice is that this is really a national network. And it's amazing when you are working on a project, you want to be PI of a new project. You cannot have all the skills and all the expertise you need all the time. But so far, at least personally, I've always been able to find the right partner within the SIB network. And it's really great because like this, you can really enjoy and work with the others and benefit from their experience. So one of the things that is going on is a mandate that SIB has received from the Swiss government, which is called SPHN, for Swiss Personalized Health Network. And what is it? Basically, SPHN is a funding scheme, so it's really funded by the government. It's intending to fund projects that contribute to make health data exit from the hospital and be used for research. So if you're lucky enough, you can have SPHN projects. Actually, I have two of them, one in oncology and one in medical genetics, but I will talk about only pathology today. And in addition to this funding scheme, there is a second initiative, and they go both very much together, which is called Biomed IT. So Biomed IT is really the technical infrastructure that is underlying the SPHN project. So it's really the IT infrastructure, the network, and all the mechanisms that go with that. The way it's organized, it's also a little bit special, but we find it quite efficient. So we have 3 nodes in the country, in different places, in Zurich, in Basel, in Lausanne. And those nodes are connected by a secure network. And what is interesting with that, if you are the lucky PI of an SPHN project, you benefit from all this infrastructure, so you don't have to do this yourself. And what it gives you access to all the procedures for users authentication, data encryption, data transfer, data decryption. And what is nice is that basically the hospitals in Switzerland, they have been also funded through this initiative to create a clinical data warehouse so that each hospital has one single point of exit for healthcare data, which is really important because it means each hospital can be onboarded once into the Biomed IT network, and when it's done, then any project can share data between hospitals. You don't need to do the same again and again. If the hospital you want to work with in your project has been onboarded for another project, it's fine, you have everything. So that's really extremely convenient and very quick. A little bit complicated to do the first time, to be honest, but once it's done, it's really fantastic because you don't do it again. So, this you know, it has been said several times also. We all know that interpretation and reporting is usually the bottleneck when using NGS. And actually, five years ago now, together with several hospitals in Switzerland, when I say hospital, I'm really talking about routine laboratories, accredited labs. So it's not CLIA. In our case, it's ISO, but it's pretty much the same. We wanted to try to understand the landscape. How was NGS used in the hospitals, and what eventually were the problems? So what we did with those hospitals, we did a benchmark, simply. We provided several data sets to all of these guys, and we said, okay, please send us back what would have been the report you would have provided to the oncologist. And actually, the results were not fantastic. We had a few discrepancies, and sometimes quite severe discrepancies. And when we digged into it, we were a little bit surprised, because we were expecting this to be caused by the bioinformatics pipeline, and actually that was not the problem. The problem was not the calling, was not the mapping, was not really technical. It was much more the individual expertise of the people when interpreting the data. And basically, obviously, there are different levels among the people, but most of all, they were not all using the same data source. And sometimes they were not very clear about why this and this one instead of this one. So really, the data source was the main problem. But when we looked at the reports and we saw discrepancies, so obviously for the patients, that's not what you would expect. So what we did together with those hospitals, we had quite a number of meetings, I must say. And after some loops of coffee and sandwiches, we said, okay, tell us, is there something we can do for you? And they came with a wish list. They say, oh, what we would like. First, we would like to have a joint repository and one repository for the country. We would like to avoid having to check 1, 2, 3, 4, 5 different resources. We would like to have one place to go to get the answer we need to put in our report. Then what we would like, we would be happy to share, but we don't want to spend time to do that. We like writing a paper because we are rewarding for that. If you post data, you are not rewarded. So we never post anything anywhere. Okay, so find a way to do this with the less efforts for them. We would like also to be sure that this is a source we can rely on. We should not ask ourselves if this is the right place to look at or not. This should be the one that we should trust. Interesting concept, they said. In a perfect world, what we would like is that the annotation that would be in this source would be ours. And all together, as a clinical community, pathologists and a few hematologists also, we would like to have a chance to approve this annotation so that we are sure if there is something in, it has been approved by at least some of us. Good. The other thing we want is if we send you data, at some point we are sure there will be discrepancies, so you have to find a way to solve the discrepancies. And we don't want to spend time for that. Great. And the last thing, also they said, well, what we don't have, would make a difference for us is a way to be notified. If we give you a VCF and there is a VUS, for example, and in 2 years from now it becomes highly pathogenic because now it's annotated, we would like to know that this interpretation has changed in this patient so that we can come back to our patient. So quite a long list of wishes. And therefore, altogether, we started to develop this project, which we call the Swiss Variant Interpretation Platform for Oncology. Also, the nickname is SVIP. And what we have in SVIP, basically inside the database, we have two kinds of informations. We have information related to the variant themselves, standalone, a variant in a disease with an interpretation. And we also have sample-related information. So this information comes from different data sources. So obviously, we use external data sources that have been selected by our pathologists, actually. We use, of course, literature and civic and ClinVar for the interpretation of the variants. And in terms of really variant information, this information comes from the VCF files we receive from the partners. They provide, together with the VCF, very, very limited clinical information. And when I say limited, it's just the disease, the gender, and the age when the data file was provided. And we receive that. Of course, we have a little bit of data management and quality control to do. And this is going through manual curation. And I will give you a little bit more details on how we do this. And what is a little bit original with this project is that then we enter in a review loop with our clinicians. So part of our clinical partners, they are members of our expert panel. I will show you also how we do that. And there is a loop, and they can complement the information we collect with their own know-how, because we have also realized, working with those people, they have a huge expertise, but it's nowhere, basically. It's in their brain, and if they leave the lab, nobody knows, and nobody benefits from this because they don't share this information. Not that they don't want to, simply it's not their core business, and they don't have time. They have to provide reports and to provide insights to cure patients. So basically, during this review cycle, they can challenge each other and they can complement what we collect with what they know. And this is really very important. The primary focus was really to support pathologists. But then we said, okay, if we do this work, at some point it has to benefit research. So this gray box, which represents this biomed IT infrastructure that I mentioned before, it's extremely strict in terms of security. We have quite a number of constraints, being technical, being legal. There is a contract with all the partners, so it's really very bound. So to avoid any kind of problems from the data privacy standpoint, we decided actually to, in a way, clone what we have inside the variant information part, so nothing sensitive at all, absolutely no link with the patient. So this is physically a completely different database, which is outside the highly secure zone, and this one can be fully accessible. There's no login, nothing, it's completely public, and it can be used for research. So we have a beacon on top of it, we have an API, or you can use also the web interface. This is a little bit limiting because, of course, inside the private instance, there is interesting information, but as a first step, we thought this is the easiest way to get ethical approval and to try to avoid any kind of a data breach. While our clinicians, and when I say our, I mean the partners who have signed the consortium agreement, and we are all legally bound by a contract, actually. In this case, all these people, they access the complete set of information directly from the secure instance. So I will show you quickly how it's looking like. So this is the landing page. Just a disclaimer, the data you will see is highly inconsistent because this is a test system so that I can share with you things that are not sensitive. So don't pay attention to the scientific or medical meaning of this information. So basically what you would do, you would search for a gene. So here you will see a gene summary that is prepared by our curators. And below you have the list of the variants that we have inside the database. If you select one of the variants, then you end up on the variant page. So the top line is a little bit of a summary. And then below, So you see number of samples 0, because again, this is the test instance. But you would see for each disease in which the variant has been seen, how many samples we have. So this is public, because the number of samples is just aggregated data. We have also the distribution of age, because for some cancers it's pretty important. Also the gender balance in the database, and a clinical significance. And I will show you how we do that. And basically, if you scroll down this same page, so we have publicly available information. Again, our partners have selected the data sources, and you see Civic, ClinVar, and Cosmic. We have the impact prediction, we show the allele frequencies, and there is something also that they were a little bit reluctant to see at the very beginning, which is the molecular modeling. And now they are starting to get used to it, and they find it very useful to support the tumor board, especially for the VUS. This is just exploratory, that's for sure, but it's giving them a hint of where the variant is located on this 3D structure. And this is a feature, actually, now they want us to further develop during our next founding scheme. So let's now, on this workflow, focus on what we believe is the original part of SWIP, which is the curation and review process. So together, again, with our pathologists, we have defined a tailored annotation process together with them. So basically, the big steps are, first, we gather public information. So we have developed a harvester, not developed, I would say adapted an harvester. And we use Varium, which is a tool I will show a little bit more in details, which is a text mining tool, actually, that was existing. It was developed, and it's still developed by another SIB group. And it has been adjusted to fit really the needs of our curators in this case. So we have this manual curation phase. And once the curators believe they have collected the right information, they push this to the review cycle, they push this to the expert panel. So we have 3 experts every time. The idea is to reach the consensus, obviously. So it might be that they disagree during the first cycle. So in this case, we have a second cycle, but we don't have a third cycle. If they disagree after 2 times, we push the annotation aside, and it's discussed live during a panel discussion. So. But this is done outside of the platform. And only once they agree on a consensus annotation, then the annotation is released. And to do that, we have 2 different interfaces on the database, a specific curation interface and a specific review interface that was developed with the curators on one side and the reviewers on the other side. So the curation interface, how it looks like, basically this is the curator's dashboard. When they connect, this is what they see, and they see 3 big blocks. The first block is the ongoing reviews, so the variants that have been sent to the expert panel. So they see where they stand. The second block is the curations that were requested, because this is a feature also we were asked for by the pathologists. They have the ability through the platform to request the curation of a very specific variant that is of interest for them. So this is what we call the on-request curation. And in the bottom, they see what their own work, basically what's going on. Because usually it's taking a while, so they can save as a draft, come back, modify, etc. And when a curator will select a variant for annotation, he will end up on this curation page. And actually, I have a video. I'm not sure it will work. I'm afraid it doesn't. But let's see. So on top, you have very general information. I will go quickly through this. The curators wanted to write down a summary. They like to do this, so they write a summary for the gene and a summary for the variant. And what you see in the bottom is really coming out of the text mining tool. The text mining tool, as I said, was pre-existing. It has just been changed. So far it's searching into PubMed Central and Medline. It's also searching into supplementary data, which is not always the case with other tools. They have added ongoing clinical trials, and they are working on adding the drugs, but this is not yet here. And basically, this is the address if you want. This is publicly available, so you can go to the Varium site. It's working nicely. In principle, they record a lot more publications than most of the text mining services. They have added also isoforms, for example. So based on what the curators want to see, this is still adjusted on their needs, basically. And when they want to curate, then they take one paper and they decide, let's go for it. So on top, there is an abstract. The abstract is there automatically. It's colored by the text mining tool so that it can highlight for you what is of interest to you. And basically, the demo doesn't work, but you have to believe me in this case. When they scroll down, what's happening is that the interface has been really designed to support the curators to make their work efficient and easy to use, because usually they complain that the bioinformatics tool is a constraint. We wanted this to be the other way around. The bioinformatics tool should be a support. So basically, they have to support the different type of evidences, and depending on each selection they make, the fields that come are conditional. If you say, I want to annotate the drug response, then the list of drugs with a peer, etc. So everything is very conditional so that they don't have to fill in 20 fields all the time. One thing that I need to mention, so we try to stick with what's going on, and actually we are extremely happy to have seen this paper out from our VICC colleagues. So far we stick to the tier levels for the annotation. They were pretty used to pathogenicity, but we wanted this to change, so we use the tier levels, but we really want now to implement the oncogenicity that is in this paper, so this is the target for our new funding scheme. And quickly, the review interface, so when the curators annotate, they annotate two different kinds of evidences. They annotate the biological significance, and they annotate the clinical significance with a tier level that you know. And for the sake of efficiency and of time, we don't send the biological evidence for review to the expert panel. That's not what they want to see. That's not what they want to contribute to. We send to them only the clinical relevance, because it's their business and we want to spare their time. So basically, if they log in, they would see something like this. So they would see one line per disease, per evidence type. And what you see in the first column, you see two fields. These are the effect that is proposed by the curators with the associated tier level. If you agree, fine, you would just say save, done. In case you disagree, then on the second column, you can change the value depending on what you believe. Then it becomes red, meaning I disagree. And if you disagree, you have to justify why. Because otherwise, you cannot enter into the review loop because the curators have no clue why you disagree with their proposal. So this is what will be the basis for the second round of discussion. And with that, just to say you can find all this information. So we have an API that is freely available. The source code is available under GPL3, so it's open source. And I would also like to highlight that it's a teamwork. So as I said, we are lucky in SIB to have different groups. So we have a group in Zurich with Daniel Steckhoven, who is my co-PI. We have the IT staff who is working heavily with us, the text mining group with another group of SIB, my group, Clinical Bioinformatics. The curation is done by the Swiss prod people. And of course, we have 10 hospitals on board, and we meet each other 3-4 times a year. And I must say, we are very happy, we learn a lot, and we believe we start to have an interesting product. And with that, I thank you, and I'm happy to take questions.
Okay, so we've got time for maybe one question, if anyone has one.
Hi, thank you again for coming over here and presenting. So my question, it really got me with the discrepancies. Did you see more discrepancies between, if you're calling it like VUS and pathogenic and likely benign, or did you see more discrepancies in the variant interpretation compared to like the actual clinical data compared to like other variants that were found? So more of like a clinical discrepancy versus like the variant interpretation of that variant?
I'm not fully comfortable with this question because due to some delays in getting all the signature of the agreement, we are not full speed, I must say, data-wise, so we are still struggling, so we are not running full speed at all. So I don't know how it's going to be when we'll be able to collect thousands of files. For the time being, it was, as long as you ask for their input, their clinical interpretation, there are very few discrepancies, usually. It was more, for the VUS, they all agree, basically. But for any reason, we had cases in the past difficult to explain. Sometimes a variant was flagged benign by one side and pathogenic by another side. It was mainly, if I remember well, it was mainly a little bit special cases where you have two mutations that come all the time together, and that should be considered one large indel and not two SNPs. And depending how you see that, then on one side it's benign, on the other side it's pathogenic. So.
All right, please join me in thanking Dr. Barbié for this amazing talk.